So, welcome back. I appreciate people taking the leap and coming a second time after the problems last week. This is lecture 2 for E443, 543. And today we're going to start doing things with CompuCell, I hope. I'm going to be talking a little bit about back-end issues with CompuCell as well, how it works under the hood. As always, I need to remind you that this class is live streamed on YouTube and made available subsequently for viewing and is lightly edited, but in general is available as it stands. I also maybe should say, and you'll see them on the slides, that many of the slides that I'm going to be using today were developed by Giulio Belmonte at North Carolina State University. These are slides that we use extensively in our summer course, and I thought I would adopt some of the improvements in the class that were made over the summer by Giulio. So today, people still haven't filled out that entry questionnaire. If people would do that, you get an extra couple of points of homework assignment. It's not a big deal, but so I get to know you a little bit better. Today, I would like to have people perhaps screen share, if they can, a little bit about their answers to homework problems number 1 and 2. I have here the schedules discussion of this week's homework problem 6, but I think we're going to defer problem 6 till next week. Not assign it this week. It makes the homework too long. Then we're going to talk about CompuCell and how it works under the hood. And then we're going to start building a single cell and seeing how to manipulate it and getting it to do the things we want it to do. And we'll run a series of exercises related to that. We'll see how far we get in class today. So we have a whole series of exercises. I'm sure we won't get through all of them today, but we could get started. So last time we were talking about the big picture, which is how do we understand biological organization? How do we build organisms? How do we maintain them? And how do they fail? How do we engineer them? And we introduced the idea of what we call virtual tissues or middle-out modeling or multicellular modeling, where we have cells. The cells do things. They live in an environment and interact with each other, and that environment is both chemical and mechanical. And then potentially they have complex control inside of those cells that steers what they do and how they respond to the outside world. For a substantial part of this class, we're not going to be putting that internal control in, because you can get surprisingly complex behaviors of these systems, even if the behaviors of the individual cells are not controlled actively. But that's always a possibility. And we talked a little bit about possible examples of what you could do with this kind of software. And we have various simulations of different kinds of dynamics. And one of the things that we talked about was that the variety of different phenomena that you can observe in these biological systems is very large, and therefore at some level you need to go down to fairly basic concepts, because you have to build an environment that's flexible enough that it can handle all of these different applications. And I should say that there are plenty of things that you might want to do in these environments that are very specialized that we don't support. But already there's a lot of diversity of the kinds of questions one can solve with these kinds of approaches. And so I guess I wanted to quickly go over, did anybody do homework problem number 2, which was simply to run the delta notch demo that was packaged with CompuCell? And if they did, are they willing to screen share for a second and share with us what they got?
All right. So I've opened the simulation delta notch from book chapters in demos folder. Here it is. There are only 2 files, a Python file and delta notch. So there's no standard XML here in this case. It's specified in Python. Okay, to open the simulation, I can click with the right mouse button and open in player. So you can also click this button here, but the caveat is that when you click that button, it will load the last simulation you loaded, it may not be exactly the simulation you want. Okay. Here I can open more windows, another window to visualize a field at the same time as the cell field. So here I have delta in the right, in the left I have the cells. So I can visualize the field delta in the right window. I can open another window to visualize notch. So I can select here instead of cell field, which are the cells, I can select a notch. And here we can see the two fields, delta notch and the cell field. You can see these white contours. This is because in my configurations, I set to show ISO values, which don't make sense here. Let me deactivate. And then now you can see, I think it only applies to one window. Let me do it for this one.
Yeah, each one has its own ISO.
All right.
At least that's essentially some graph player.
And there we go.
So there was a good question, which is why does it produce a hexagonal grid? And the answer is that there is what's called lateral inhibition going on. That is that the cells, each cell that is trying to be high delta and high notch and trying to force its neighbors to be low delta and low notch. And if you try to look at self-consistency, you go around a circle. If you have three cells in contact with each other, you can't have, if A is high, B was low, C can neither be high nor low, right? Because it's going to mismatch one of its neighbors if you have three adjacent. It winds up that depending on the detailed settings of that system, you could get situations where you get oscillations. Or if you weight it in favor of could be in favor of uh, inhibition, then you get a situation where your high one is able to win out over all of its neighbors. And so if you have isolated high spot surrounded by all low, that could be self-consistent. So you have lows, every low cell is next to one high cell or two high cells. Every high cell is next to only lows. And you would notice if you looked at Pedro's simulation in a little more detail that there are a couple of places where the arrangement isn't a perfect hexagonal lattice, and those cells were confused. They were fighting each other over who got to be high and low. And so if you did, say, a square lattice instead of a hexagonal lattice, you might get some different things. In reality, the cells can have rather complex shapes and interconnections. And in that case, you actually do, in biology, could get oscillations where there is no stable pattern. And our friend Lorenzo Veschini at University College London, I'm sorry, King's College London, does experiments and simulations of that oscillation pattern, making dynamic measurements of delta and notch in living tissues, not in vivo, but in explanted living tissues in culture.
So my question is, if I do a quick search about delta notch on the internet, I will find that either delta is high and notch is low, or notch is high and delta is low in one cell. So I would, in a two-cell system, I would have high, low, low, high, instead of high, high, low, low, as you mentioned. So why is it the difference? Why in the simulation we get high, high, low, low? And when I search, for example, in Wikipedia or in YouTube, they will say the configuration would be high, low, low, high.
The question was, why are delta and notch both either both high or both low in the simulation that we wrote? Whereas in biology, typically you have high delta, low notch, or high notch, low delta. The answer is twofold. First, the simulation itself uses a very simplified approximation of delta notch. And that simplification omits some complexities of the way the signaling really works. The second is that you can get that simulation to do the more biological result where you have the opposite phase if you tune it, if you play with the parameters. So and biologically, sometimes you can get what you see here. So these systems where you have essentially it's a reaction diffusion system with a long range inhibition and relatively short range activation. It's possible to set those up so that you either get the activator and the inhibitor both high in the same place, or they go opposite to each other. And so it's a good question. You're right that biologically the high notch, low delta, low notch is more common than having them vary together. But both do happen. And you could use that in simulation and explore those variabilities. That kind of thing is discussed, if you're really a mathematical biologist, that kind of thing is discussed in some detail in the later chapters in the second volume of Murray's Mathematical Biology. Although I don't think he specifically does delta notch, but he does this question of when do you get covariation versus opposite variation in the reaction diffusion systems. And there are certain mathematical constraints about the pattern of cross inhibition that determine that. Okay, did anybody run the cell growth and division simulation and want to show it to us?
So the first thing that I did was I ran the simulation with the default parameters and then just took a screenshot at the first time. So I'll share that. So that was what the initial configuration looked like. Shortly after initialization, things had begun to grow just a little bit, but not enough to notice anything significant. So then I ran it for 150 steps with those default parameters, and this is what I got. So this would be the baseline comparison to check the growth and like the cell size against a bunch of different parameter situations. So then I tried a configuration where I let the cell type medium have like a higher adhesion energy than the other cell types. And I think there's a little bit less growth than in this default configuration. This center region is a little bit bigger. There seemed to be maybe a smaller band here, although that could just be coincidence from running the simulation again, because there's a lot of variability between runs. And then I made them all have a higher adhesion energy. I think I changed it from 10 to 20. I think that was it. Maybe it was 50. It looks smaller, generally, more square than the default at 150 steps. Then I made the adhesion energy 70 for all of the cell types, and that's clearly much smaller. It doesn't look much different from the default. The next thing I tried was I reset the adhesion energies, and then I increased the growth rate. So I increased the volume added to the cell each run from 1 to 5. And by 150 steps, the cells had just completely invaded the little square. I think, so there's no region in the middle with these larger cells. I don't really know why that is because I don't understand the details of the simulation. There might be more variability in cell size, although can't really tell visually. And that made sense to me. Okay, if you make them grow faster, there will be more of them. at step 150. So then I increase the volume at which they had to, well, I increased the volume they had to reach to divide from 50 to 150. And they are all bigger, and there was less growth comparatively than at 150 steps with the default parameter. So no surprise there. And then the last thing I tried to do was I wanted to see if I could make an increase in the division volume balanced with an increase in the growth rate. And I don't really know. I mean, I don't think they balanced out, because when I multiplied each one by 3 and then multiplied the axes by 3, the area taken up by the growing cells was smaller than in the initial one. So maybe it's not multiplicative. Maybe they don't cancel out. I would have to test a lot more values.
Okay, thank you. That was fantastic. You actually introduced a lot of the things we're going to do in class today, so it couldn't have been a better introduction to what we're going to cover today. So now, having played with it a little bit, I'd like to tell you a little bit about CompuCell3D. We'll talk about how cell behaviors, cells and their behaviors are represented in CompuCell, how they move, and how we specify these things in a simulation. And we've done two very, very simple examples, but we've seen some very complex ones. The fundamental back-end things that we're doing are the same. So we start with, say, a picture of some cells. These are epithelial cells. I think they're probably MDCK cells, although I'm not sure about that. The red is stating for probably cortical actin. The green is stating for the nucleus in this case. And we see hexagonal or roughly hexagonal cells, polygonal cells. This is a projection. The cells would be in a flat layer in an epithelium. But here's a typical experimental image that we might start with. And different simulation methodologies are going to make different choices about how cells behave and what properties they have. So one way we could represent cells would be to put a lattice down and have each voxel of the lattice represent a single cell. And so every cell in the lattice would either have a cell there or no cell there or different cell type there. Clearly then every cell is a square. Every cell is the same size. Cells can't move continuously in space. On the other hand, we could certainly put a lot of cells together. And so that is how cellular automata models work. And so that gives us, in a sense, the fastest computation, but also the least spatial granularity and the least flexibility in terms of what the cells do. Another way to do things is to use what we think of as more of a molecular dynamics approach, where we say we're going to specify the center, maybe the nuclear position or the center of mass of every cell, and say the cell behaves like roughly like a spherical object with some radius of interaction. And again, the molecular dynamics code is extremely optimized to solve these kinds of problems. So you could use great back-end solvers like, well, there are many molecular dynamics solvers. But the cells don't intrinsically have a shape. They're really just positions and radii. Now, it's possible to make the cells ellipsoids and things like this, but to give more complex shapes becomes problematic. On the other hand, there are a lot of nice things about not being on a lattice. You still have to use lattices for chemical fields, but that's a different matter. And Physicell, Paul's package, uses this choice. TissueForge, TJ's package uses it. Biocellian uses it. There are a lot of packages that use what are called center models to represent cells. In this case, the position of the cell is well defined. Cells can move continuously. On the other hand, there is no surface, for example, of the cell. There is no shape of the cell. The cell doesn't even have really a volume associated with it by itself. Another thing that you could do is, if you look at this picture, you could say, well, what really is striking me about this picture are those red edges of the cells. And those red edges actually represent, as I say, the actin cortex. They're fibers inside of the cell that are fairly rigid, and that also are under tension. They're pulling. And so you might say that really what's going on in this tissue is dominated by the behavior of those actin fibers, those red fibers. And so I'm going to represent this pattern by putting spots at the vertices. In this case, it's 2D, so threefold vertices, and essentially springs along the edges. And I'm going to adjust the behaviors of these springs. Are they longer? Are they shorter? Are they under more tension? Under less tension? You can do this in 3D too, although it gets a little bit complicated to do the bookkeeping when you do. And this is called a vertex model. And this kind of model is actually very powerful if you want to understand reorganization of cells within an epithelial sheet, for example. And people, for example, in the community who do Drosophila, fruit fly development, use vertex models a lot. And so this is a rather powerful technique, and it has its own set of computational tools and approaches. Notice in one case, the cell is a point object that has no volume or surface. In another, and the position is on a grid. In one case, the cell has a point object which has an effective volume but no surface. It's off-grid. Vertex model, the center of the cell is a consequence of the position of the vertices. We have to infer the center position from the edges. And the volume is actually rather complicated to determine. And so these are rather different sets of possibilities that we could use. So here are three different approaches to modeling the same picture. And we're going to have to make different measurements and different decisions to be able to turn that single experimental image into a computational simulation. In our case, we're going to be using a lattice method, known as GGH or cellular Potts model. In this case, we do, in a sense, the naive thing, which is we say all of the voxels in our photograph belong to cell number 1, so we label them 1. All of the voxels in this region belong to cell number 2, so we label them 2. All the voxels in cell number 3, we label 3. And so we go directly from the voxel image of the experiment to the voxel image of the simulation. Of course, we may make the simulation resolution maybe a little bit higher or lower than the one in the experiment, but conceptually, we're mapping a voxel image from an experiment to a voxel image in the simulation. This gives us more spatial resolution. It gives us a little bit more flexibility than some of these other techniques. It has some limitations. Being on a lattice is going to have some artifacts that we have to deal with. We still don't have the membrane of the cell represented as an explicit object, which becomes an annoyance when you get to complex models. And so each one of these methodologies has some advantages and disadvantages, which is one reason that there's so many different modeling packages out there, because they implement these different approaches. One thing we're hoping to do in the future would be to have a way of specifying these models that's independent of the methodology. The problem is that because of the way you characterize the cells is different in different methodologies, it's a little bit hard to define a method-independent model specification. Okay, so we now have a concept of a cell, which is a set of voxels in space that say I'm part of cell number 1. And the next thing we've talked about is that biologically cells move. And so we have to have a rule for how cells move. And that rule is going to seem, if you haven't come from a statistical mechanics physics background, this rule may seem rather artificial. I'm not going to dive into the physics of exactly why the rule has the form it does, although I'll give you some assertions about it. But what we're going to do is the following. We're going to, at each time step, micro time step, you've seen in CompuCell there's something called the Monte Carlo step, which is the fundamental time unit in the system. A Monte Carlo step is composed of thousands or even millions of micro time steps, and the micro time steps have a particular and perhaps strange looking process, which is here's our cell at initial time. We pick one site in the cell. We pick a neighboring site and we ask the question, if the cell extended itself, if the cell we've picked were to extend itself and push itself out by 1 voxel, what would happen? So in this case, the cell gets bigger by 1 voxel, the neighbor gets smaller by 1 voxel, the boundary gets longer, the center of mass moves a little bit. And so we're going to say that there's going to be some penalty function. If you're used to loss functions in neural networks, you're used to this kind of idea, where there's some loss function or penalty function, we're going to have a little bit different name for it. And if our move improves our situation, reduces our loss function, we're going to make the move. If the move makes the loss function bigger, just as with neural networks when we're training them, we don't necessarily reject the update. We'll accept that update with some probability that decreases with the cost penalty of the move. And the reason we do that has a good statistical mechanics background to it. There's a lot of physics under the hood there, but that's fundamentally what we're going to do. And so then in this case, we accepted that first move, and now we're going to pick another lattice site, and we're going to try to copy that one, and we'll try again. So we're going to have, each time we'll pick a lattice site at random at a boundary. We'll ask the question, if the boundary extended to one of its neighbors, what would happen to the penalty function or loss function? We'll roll the dice and either accept or reject that move, and then we'll do it again. The result of this is that many of the things that we try don't happen. We spend a lot of time rejecting possible moves. The movement is stochastic. So in a sense, this seems like a very strange thing to do. It comes from a branch of statistical mechanics that goes back actually to the Manhattan Project, when they were evaluating what are called partition functions in statistical mechanics. And this particular algorithm we're using here is a modification I developed of what's called the Metropolis algorithm, which was used to evaluate equilibrium statistical mechanics properties. But it has the advantage that it does actually give you the kind of movement on average that you want, and we'll come back to that in a second. Now, I've talked about this penalty function or loss function. Because of the history, we call this an effective energy. And for historical reasons, physicists like to write effective energies as H's, although often they will write E instead. And the advantage of this is that we can write a term that says the volume of the cell is approximately this. The surface area of the cell is approximately this. The length of the cell, there's a typo there I see, is approximately this. The contact area of the cell is approximately this. And we can add all of those terms up, and then the dynamics that I've just specified will automatically try to evolve the pattern to satisfy all of the constraints and conditions that we've specified. And we'll come back in a few minutes to what those constraint functions look like mathematically. Any questions about that so far? I generally try to not do too much math or physics in this course, but I thought it was worth at least spending a little bit of time trying to introduce some of the basic ideas. You don't need to understand these things to be able to build models, but you'll have an easier time tuning your models and also diagnosing what's wrong with your models if you have a basic understanding of how the internals of the system work. And so now we have this idea of a loss function, and we're going to dig in a little bit more. When we make our copy attempt, we have a loss function, an energy function value before the copy and after the copy. And so if the copy happens, the energy of the system changes. And the copy is a move of the boundary of the cell over a distance, in this case, 1 lattice site. And an energy divided by a length is a force. And so basically, the change in energy when we move the boundary tells us what the force acting on the boundary is at any given position. And so the energy formalism that we're using here is actually equivalent to a force-based formalism. We're saying that we're going to measure the force acting on the boundary given all of the different processes the cell participates in at any given time.
Does boundary mean boundary of the cell or of the general region occupied by? Like at what level is it?
Okay, so the question was, what does boundary mean here? Well, if I pick a point in the middle of my cell here that's blue, and I try to copy blue to one of my neighbors, the configuration doesn't change. And so I reject that attempt because it's not doing anything. So in this case, boundary really means the boundary between two cells. Now, if I had an internal structure to a cell that was blue, then of course I could have movement within the cell as well. But in this simple model that I have here, where the cell is essentially a bag, a featureless bag, the only way the cell can move is by extending or retracting its surface. So that's a great question. And there are some things about this kind of modeling that are a little bit counterintuitive. Again, the surface of the cell is very important, but there isn't an object at the surface. The volume of the cell is an object. The surface of the cell is simply the voxels that are which are next to a different kind of voxel, and that has its own set of consequences and annoyances, which some people like Morpheus software package actually try to get around. Maybe CompuCell needs to be extended in that way. Good question. So in this case, I'm going to say that if the force is pushing in the direction of the copy, I'm trying to move in a given direction. If the force on the cell boundary is pushing in the direction I'm trying to move, then I move. End of story. So I accept a copy attempt that lowers the pattern energy with probability 1. Now you might say if I'm pushing uphill, I shouldn't ever accept the pattern, the update. But in fact, I'm going to accept the update with a probability that is exponentially decreasing in the amplitude of the penalty. And you could say, why would you do this? And in fact, it's not clear that the exponential form is really the right one to use. But cells are not passive objects. They're what are called active matter. That's why cells are alive, one of the properties of their being alive. And they move actively. And those actin filaments that we saw in red in that experimental image are actually little motors that are actively pushing on the cell membrane. And so the cell has a series of little motors inside of it that are continually pushing and pulling on the cell membrane. And so there is force generation inside the cell at all times, random force generation inside the cell at all times. And that force generation can be enough to overcome the force opposite to the move that I'm trying to do. You could say, well, why use an exponential cutoff instead of a linear cutoff, for example? One can actually do a linear cutoff in CompuCell if you want to. An exponential cutoff has the advantage that it goes from 1 to 0 smoothly, and so it's mathematically a convenient functional form. It also, in terms of statistical mechanics, has certain convergence properties that are appealing. But fundamentally, the idea is the following. If I'm trying to move in a given direction, if the system is pushing me in that direction, I do. If the system is pushing against me, I ask the question, is there enough energy from the motors inside of me to overcome that obstacle? And if so, I move in that direction anyway. And again, if you've done neural network training, you know that you don't only accept updates that reduce your loss function. You allow updates that increase your loss function a little bit, actually usually with the same exponential probability. Seems sort of funny to be using neural network learning algorithms as a justification here, but for different backgrounds, that may or may not be a helpful concept. One result of this is that if I ask the question, if I look at the net velocity of a boundary, at the time step 1, the boundary moves forward. At time step 2, there's a probability it moves back where I started. And so I could say with this jitter forward and backward, what's the net velocity of a boundary? I discover that the net velocity of an object or boundary is proportional to the gradient of the effective energy or the force. And so this algorithm is noisy, it's stochastic, but it does have the result that the velocity with which something moves is proportional to the force applied to it. Now, as a physicist, you learned that Newton's law that the acceleration of an object is proportional to the force. But at the scale of individual cells or boundaries of cells, we're in what's called an overdamped regime. Friction and viscosity are very, very large. And so the velocity actually at the cellular level, there's no inertia. The velocity of objects is the force divided by the drag on the object. If you stop pushing, they stop moving immediately. And so velocity proportional to force is actually what you want in the regime that we're operating in. It's actually possible to extend this kind of method so you get inertia, but you don't get inertia for free. You have to add it to the system. There's another thing that happens, which is this linear force velocity relation, which is what you want. And this is a simulation we could do in class, maybe next week, if people are interested. It works if the forces are reasonably small. If you push too hard on things, they break. And it sets what's called a speed limit in the system, which is that if you try to push cells so that they move more than about 1/5 of a lattice site per Monte Carlo step, the system will not behave well. So there's an artifact there. Okay. So that's the dynamics of the system. And again, there's this rather complicated statistical mechanics under the hood, but from the point of view of actually how it works, it has a simple result. If I push on something, the velocity with which the thing moves is proportional to the force with which I push. So it has a simple consequence. Now, we've talked about constraints and energy functionals. Again, if you're coming from a physics background, the idea of what's called an elastic constraint will probably be familiar to you. If you're coming from elsewhere, again, if you've done neural networks, these are going to be things that you'll have used, although they may not have been described with exactly the same language. And so, for example, suppose that I say that I want the volume of my cell to be V target. As I mentioned before, I cannot enforce that the volume of a cell is something because the cell does not exist in isolation. If I took a bunch of balloons and have some volume sitting here in this room, and then I stuff them into a box, they'll be compressed and the actual volume per balloon will be smaller. And so I could say that the balloon has a target volume. It has a volume when it's sitting in the room without anything happening to it. But if I push on it, it will get smaller. It'll be compressed. And similarly, if I put the balloon in a vacuum, the balloon would expand because the volume of this balloon is dependent on the pressure acting on it. And so we have what's called a volume constraint, which says that the volume energy of the cell is a compressibility factor, lambda volume, times the actual volume of the cell minus the target volume squared. So that energy is 0 if the actual volume is equal to the target volume. If the actual volume is bigger or smaller than the target volume, then that contributes a positive energy. So this creates a force that pushes the cells to have the volume that they're set, their target volume, but doesn't absolutely force them to. The bigger lambda volume is, the more closely the actual volume will track the target volume. And we're going to play with this in a minute, make it less abstract. The same thing with the surface area of the cell. How much perimeter does my cell have? I can't specify that exactly because I don't know what the environment of the cell is, but I can say that the cell has a certain amount of membrane, like the area of the balloon itself, and I can pull on it or push on it. And what I see is the actual membrane area will depend on the constraints on the system. And so you could say, why is it what's called, why is it quadratic or elastic? Why is it V squared that I see? And the answer is that the force is the derivative of this. And so the force of V squared, the derivative, the first derivative of V squared is V. And so this is equivalent to something like a spring. It's equivalent to a linear elastic spring. And so the further away I am from my target volume, the force, restoring force, is proportional to the difference between the actual volume and the target volume. And that's exactly how the ideal gas law works for a balloon, for example. And so quadratic energies give you linear forces. Again, if you're not a physicist or you haven't run into this, I wouldn't worry about it too much. But when you come up with pathologies in this kind of simulation, having a little bit of this background may help you do some debugging. So suppose that the current volume of the cell is V, and I now make, and the actual volume is smaller than the target volume. And now the cell tries to extend itself by 1 voxel. Well, my volume, actual volume has increased, so my energy penalty has gone down because the difference between the actual and target volume is decreased. And so in this case, the volume constraint is favorable for me, and I'm going to tend to accept that update. On the other hand, if the cell were already bigger than its target and I tried to extend it, now the energy is going to go up, the volume energy is going to go up, the penalty will go up, and so now the volume energy is pushing against extending that surface. And we'll play with that and see what it does in just a minute. How far we allow variability from our ideal will depend on that lambda volume term, and it will also depend on that fluctuation amplitude. If the motors in my cell are really strong, they can overcome relatively strong obstacles. If the motors in my cell are fairly weak, then they won't be able to fight against strong obstacles. And so the probability of an adverse move is going to depend on the fluctuation amplitude, which has the unfortunate name of temperature in CompuCell, which I regret bitterly for having adopted from the physicists back 30 years ago.
What would you call it?
Fluctuation amplitude. Or fluctuation energy, because it's really an energy length scale. Let's say energy length scale. It's an energy scale. But the physicists use T, the character T, capital T for temperature, because it's derived from physics calculations in which T really was a temperature. Originally, when these ideas were developed in statistical mechanics, molecules were moving. Yeah. And so we have to get used to that kind of result. One thing that we'll find is that we can have many forces acting on the cell at the same time, or many constraints acting on the cell at the same time. We can't necessarily satisfy them all at the same time. In the homework, we'll ask you to evaluate by hand a couple of very simple cases to get a feeling for that. So for example, suppose we said that the volume of our cell wanted to be very large, our target volume of the cell wanted to be very large, but the surface target was very small. That would be a little bit like a golf ball. Maybe golf ball is the wrong example. It would be a little bit like a soccer ball, where we're trying to push a lot of air into a small perimeter volume, and so the cell will get very stiff and round. the flip side is, suppose that the target volume were small and the target surface area were very large compared to that. Well, that would be like an underinflated beach ball where we got a lot of membrane, but there's not very much air inside. So if you want, you can think of the V target as the mass of the cell. And you can think of S target as the amount of membrane the cell has. And clearly, there's some regimes in which the cell is relatively round and under-constrained. Some regimes in which it's very floppy, some regimes in which it's very stiff. And so we can have multiple constraints present. When there are multiple constraints, we can't satisfy usually all of them at the same time. There's a little bit of an extension problem in the homework where I ask you to determine that analytically for a simple case of a circle and a sphere. One can, if you're not on a lattice, but you imagine just essentially a beach ball in the real world, you can actually calculate exactly what that competition looks like. It's not a requirement because it requires a little bit of the algebra is a little bit messy. But, if you feel like it, it's a calculation you could do. So let's just see what happens with fluctuation amplitude. Let's see two simulations here, two simulated cells. And can somebody tell me which one has a higher fluctuation amplitude of these two movies? Kyle says the one on the right has a higher fluctuation amplitude. Yes, the boundaries are more broken up. You're seeing more random movement. Now, you might well say the one on the right is probably not realistic. The cell seems to be falling apart and reforming itself. On the other hand, if you squint, maybe the cell really actually is spreading out and trying a lot of things over the time scale over which you're looking. So I can't say absolutely that which one is right or wrong for a particular simulation, but you can see clearly that as we change that fluctuation amplitude, the amount of fluctuation at the surface, the degree to which the cell is broken apart, will change. If I put lots of cells together, and I don't think we'll get to this example today, and actually you mentioned seeing this effect where you change the surface energies and the cells got rigid and didn't move very much. What matters is the surface energy relative to the fluctuations that determines the degree of motility of the cells. And so here you're seeing exactly that. On the left and the right, they're different. Which of these two has the higher fluctuation amplitude? And there's a bit of a trick in this picture. First, a speculation about which has higher fluctuations, and secondly, what's the trick? If I look just at the blue cells, which one has more surface fluctuations, the right or the left? The right. If I look at the green cells, which has more surface fluctuations, the right or the left? The left one has more surface fluctuations. The right one, you can see the surfaces are lining up along the lattice, and they're not moving that much. Because what really matters is the ratio of the energies to the temperature, to the fluctuations. And in the right-hand one, I've set things up so that the green cells have very high energies compared to the fluctuations, and the blue cells have low energies relative to the fluctuations. And so that's a pathology that you're going to run into later on in life in this kind of modeling. And it's something that we'll return to periodically, but you're seeing it here. The one on the left, I would say, is relatively well behaved. The cells have some ability to move. Their boundaries aren't too broken up. The one on the right, the blue cells are falling apart completely, which is not usually biologically meaningful. And the green cells are basically square and not moving very much. And that's probably not biologically meaningful either. Certainly, there is no lattice in the real world, and therefore, if the boundaries could be stiff, but they shouldn't be lining up on the square like that, which doesn't exist in the real world. There certainly are situations in biology where the boundaries of cells are pretty stiff, for example, in plants or in bacteria. And so you might want your boundaries to be fairly rigid, but you don't want them to be lining up against on a lattice, which doesn't exist in the real world. Okay. And so again, the moral here is that it's the ratio of the effective energies to the fluctuation amplitude that determines the behaviors of the cells. And we're going to learn in the rest of this class to begin to think about how to set up cells that do what we want, that have the right amount of fluctuations, that have the right behavior. Well, right is maybe not the right word, that have reasonable behaviors. We want cells to have roughly stay connected, not have too many stray pixels or fall apart into separate domains. Boundaries should be fluid but not completely crumpled. They shouldn't line up on the lattice. Cells should be more or less round. Now, sometimes they're biological. One thing about biology is almost every rule has a counterexample. 99% of the time it's this way, but once in a while something strange happens. And so it's not that these things never happen, but we have an idea of sort of typical cell behavior.
So typical is the word. Right?
Typical is a good one, yes. Kyle points out typical is a good one. And so it's also, but it's very useful, I think, to be able to have already from the beginning an idea of, gee, if it looks like this, something's wrong, and an idea of what to look for. Now, these are not syntax errors in your code or type or failures of the code. These are how you pick the behaviors that are reasonable for the physics and biology of the system you're looking at.
Is it typical for the physics or how it displays equilibrium?
So Kyle says, is it typical for the physics or how things display equilibrium? The systems we're looking at are never really equilibrated because we always are putting energy to the system through the fluctuations. But if things freeze, if the boundaries of our cells freeze, that's typically not biologically realistic, although, again, there are situations in which cells do things where the boundaries stop moving, so it's not impossible. They can, again, once we start playing with this, and I hope it'll make sense, but I want you to go over these ideas before we play with them, so that when you start seeing these things happen in your simulations, you'll have an idea at least of, gee, we talked about that. One thing that CompuCell does that's an option that we rarely use is that fluctuation amplitude tends to be set globally in the system. And then you adjust the energies for each cell separately to make the cells behave properly. In principle, you could have different fluctuation amplitudes for each cell, and that's allowed in CompuCell. It gets to be a little bit complicated to make it work. And so people tend to stick with a global fluctuation amplitude and then play with the energies. But it's rare, but if at some point, if somebody might want to understand how that works. Okay. Okay. So here, if the fluctuation amplitudes are too small compared to the effective energies, we get things like those green cells on the right, where the cells don't move very much. The cells' walls line up with lattice directions, and they have sharp corners. If the fluctuations are too big compared to the effective energies, you get things like the blue cells on the right or that red cell towards the left, where the cell splits into multiple domains. We have lots of stray voxels and the boundaries crumple. We'll find that there are pretty big ranges of parameters which things behave well or reasonably. But you do have to be a little bit careful about this. It's not done for you. You have to be aware of that. Okay, time definition. We have a concept of a force and a concept of velocity, because we can say that the scale, if we know that our cell is 10 microns across and we have 5 voxels across, then 1 voxel is 2 microns. So length scales are unambiguous in CompuCell. Time scales are a little bit more complicated, because in an overdamped force velocity relationship, the velocity is the force divided by the drag. And the drag in this case is not something that we specify externally. It comes from the dynamics. It comes from the way we do our updates. And so there's a little bit of ambiguity about the timescales in a system like CompuCell. And it may sound funny, but you can actually pick any timescale you want as long as the time assigned to per Monte Carlo step is short enough. Essentially, there's a speed limit. As long as you obey the speed limit, you're all right. But 1 Monte Carlo step is clearly defined operationally. If I try to move as many boundary voxels as there are lattice sites in the system, that's 1 Monte Carlo step. Now, it's not the same as actually trying to update every lattice site in the system, because I pick the lattice sites at random. And so some lattice sites may be picked twice in a Monte Carlo step, some may be picked not at all. But on average, I'm trying to pick each lattice site once. Now, in principle, one could come up with different lattice site picking rules. For example, you could have a list of all the sites, and you could randomize the order of that list. So you force every site to be picked once. And that will also work. It's computationally a little bit more expensive, which is why we don't do it. The one thing that you can't do is synchronous updating. If I pick lattice site 1 and then lattice site 2 and then lattice site 3 and sweep through the lattice in an orderly way, I get total garbage from this algorithm. It doesn't converge. And in fact, the basic algorithm that we're using here is extremely good test of the quality of your random number generator. If you use a simple random number generator like the old Fortran RND or the old C RND function, and you run this, the pattern of the cells will be totally ridiculous. It's actually very, very sensitive to correlations. The fact that things move correctly depends on the choices being uncorrelated with each other. And so if you want a random number generator test, it's actually a pretty good one. You have to use pretty good random number generator to get this to work. But 1 MCS represents any time less than the maximum time, which is that you don't want things to move too fast per Monte Carlo step. If you push cells too hard, they'll break or teleport, and you don't want that. Again, we'll come back to this, but I'm giving you there's always a question of, do you teach vocabulary first, or do you teach grammar first, or do you teach conversation first? You need to learn a little bit of vocabulary and a little bit of grammar to talk, but it's not very interesting to just do grammar by itself. And today we're doing a little bit of grammar. This is essentially grammar. And I hope that having heard the grammar once, then when you start playing with and talking, it'll make more sense. We can always come back to it. Operationally in CompuCell, there is a very simple time slicing algorithm, which can be called operator decomposition or various fancy names. But essentially, it means we move the cells with everything that's not a cell frozen, and then we do the other things. And what are steppables, we've heard the word steppable already once or twice, are called steppables because they run in between the Monte Carlo steps. Another bad name that we would never have kept if we had known that it was going to be used for 30 years. And in fact, CompuCell lets you run the steppables more often than once per Monte Carlo step. with an even worse name, which is called flip to dim ratio. But in any case, to
List.
our list of badly chosen names in CompuCell. However, the basic idea is the following. Suppose that I have chemical fields in my system. What I do is I say the chemical fields are such and such. And then I update the cell positions, assuming the chemical fields are frozen. And then I update the chemical fields, assuming the cell positions are frozen. And then I update the cell positions again. So I alternate the solvers that I use. And that works provided that the time steps are fairly small. If you try too big time steps, that becomes numerically unstable. But trying to do something fancier tends to be impractical. And so when you have stepables, they will run in between the movement steps of the simulation. Okay, so what I'd like to do now is start actually building some simulations. And we're going to learn how to build a cell, one cell, and we're going to then learn how to control it. Basically, we're going to do that top one, that red example. Cells, in fact, come in a wide variety of shapes and sizes. We've already talked about the what are called epithelial cells in a layer, which are rather, if you look from the top, are rather polygonal. There are also what are called mesenchymal cells, which are more blob-like or amoeboid, typically. And then there's some cells that are of interest that have special shapes and properties, like neurons or podocytes that have rather complex shapes. But your generic garden variety cell typically is either epithelial or mesenchymal. So I'm going to walk through the steps, and I can walk through them. Does everybody have CompuCell running? I'll walk through this once on the screen, and people can try to follow along. Then we can go back and do it again together if people don't get through it. So what I'd like people to do, I hope everybody now has 4.5.0 running on their computers, so we don't have the glitches that we had last time. And I noticed there's already a release 2 of 4.5.0, so somebody must have caught a bug. So Maciek has been very fast in updating that new version. I'm still running 4.5.0.1, but that should be all right for today. So go into Twit and try to remember from what you did before. Launch Twit, a new CC3D project, and give me a thumbs up when you're ready to do this, because it's going to be pretty boring if people don't follow along. Or if you like, I can go through it and then we can come back, circle back if you want. I can walk through all the steps. Maybe I'll do that first. So you're going to go to CC3D project, new project, and then you're going to give it a name. And you can see this was from the 2021 workshop that Julio did. In this case, we called it module 1.3.1_3_1_EMT, but you can give it a name like simulation 1 or exercise 1 or 2_1. Don't use a point in the name. And say Python plus XML as an option. That's the default. Okay. And we're going to leave the defaults for the lattice size and everything else as they are. But make sure that for initial cell layout, you select rectangular slab, not blob. And we'll set continue. You can tell Julio uses a Mac, huh? So the screenshots are all Mac screenshots. If these were my slides, they'd have Windows screenshots. You'd be able to tell the difference because the windows themselves would have slightly different layout. And then we're just going to create a single kind of cell in the next window where we cell type specification. We call it cell 1 or my cell. And you have to remember to hit the add button to create the cell type. And again, don't put spaces or special characters in cell names, and don't start them with a number. That may seem obvious, but I promise you that people do it. Okay. And we're not going to have any chemical fields, so you hit continue in that window. And now we are going to have the main window that we use to set what kinds of things the cells do. And we're going to select contact under cell behavior. We're going to select volume flex and center of mass. And that's it. Given what we're going to do in a few minutes, it might actually be useful to hit surface flex too, but don't do that for the moment. And what this is going to do is each one of these cell properties and behaviors creates one of those energies, those effective energy functionals that I talked about. So when we select contact energy, that means that there's going to be an energy per unit surface area, which we'll come back to, of the cell. Volume flex means that there is some target volume for the cell and some compressibility. Okay. And now you hit done. And I hope that now in 4.5.0 it doesn't crash. So please give me a thumbs up if this worked. And if people would like me to step through this again, I can. Or you can follow all the steps here up on the screen. So why don't you see, if you didn't get it all done while I walked you through it, why don't you look at the steps 1 through 7 and see if you can get through it on your own. Okay. So now we're going to talk about what's inside of the simulation. The simulations are made up of 3 or more files, typically. Sometimes there'll be only 2. Sometimes there'll be more. But the default simulation structure that we built will have 3 files. And this is both a strength and a bit of a weakness of CompuCell. It's a strength because each file does something specific. It's a weakness because there isn't a unitary file which serializes all the information. Sometimes it's nice to have the whole simulation be in one flat file. And there are some languages that do that. So we will have an XML file, CC3D that specifies parameters and the static properties of the simulation. Although in many cases, those properties can be overwritten. And we'll come back to what those are, what those static properties are in a little bit. We have 2 Python files, one of which is called main Python script. And we will not use that very often. When you're doing advanced things like optimization and running on clusters, for example, you'll use that main Python script more. But typically that main Python script is what's sequencing the calls to the various solvers. And that's typically built for you automatically. You don't have to deal with it. And then there's a third file, which is called Python steppables. And that one is we're going to be using a lot. If we look inside of the main Python script, it's gotten simpler over time. It used to have a lot more Python boilerplate. From CC3D, import CompuCell setup. From module such and such, import steppables. That's the things that actually do the work. Register steppables, and then run it. So it runs the simulation. So CompuCell fundamentally is a Python script. The Python steppable is where we do the things that we want to do. If you're interested, well, we have a lot of classes, Python classes. Again, the fact that that your behaviors and things are specified as Python classes is sometimes of utility to you. For example, you could load other kinds of classes into CompuCell if you want to. If you're not familiar with those aspects of Python and classes, you can ignore them for the moment. But you'll see that the steppables import a steppable pattern. And then we have a class, which here is something steppable, which inherits steppable base pi. And then inside of that steppable, we're going to have a variety of functions, some of which are always there. The init is something that we have to have inside of a class. The start, step, and finish, and now on stop functions are things that we're going to use as standard structures for how we define our models. And we'll come back to that. But the init is something that creates the steppable for us. The start function is called once at the very beginning of the simulation. The step function is called periodically through the simulation. The finish function is called if we run out of time. If we say the simulation runs for 1,000 Monte Carlo steps, we get to 1,000 Monte Carlo steps, the finish function is called. And we might use that, for example, to export the results if we were running this in a batch mode. And then on stop, which was added fairly recently, is called when you hit that stop button on your player screen, because that's a different exit, technically a different exit from CompuCell from running out of time. Although often you'll do the same thing in either case. Okay, so remember we have three basic modules that specify the simulation. The main Python file, which we typically won't hand edit, although we can. The XML, where we define what the plugins and energy functionals are and set parameters, certain kinds of parameters, global parameters. And the steppable file, where we actually do the dynamic, flexible things in our simulation. Okay, so I'd ask people now to run the simulation that they just generated, and you should see something like this. And remember, we have those blue buttons. We have the play button, the single step button, the pause button, and the stop button. And did that give you something like this when you ran it? Did it run? It ran. Good. So unlike last week, CompuCell's working now. That's good. Okay. A lot of things are happening in the simulation. Let's focus on just a single cell and trying to understand what it does. So we're going to now work on editing the simulation. And to do that, we need to launch Twit. And so to launch Twit, there are a number of things that we could do. Do people have Twitter available on their desktops? Everybody? You use Twitter to generate the simulation, so you should have it. The one thing you may have to do is the simulation that you have may be compressed. And so here under CC3D project, you may have to double click on the name of the simulation to open up the tree of components in the simulation. Okay. And that should expand to 3 tabs. You'll see simulation.xml, simulation.py, which is that main Python script, and then simulation.steppables.py. Does everybody see that? Alex, you got that? Anybody not see that? Okay. All right. So now we're going to change the initial arrangement of our cells. Double click or select the tab for the XML module, whatever your file name is, .xml. Open that one. And it'll say CompuCell 3D revision something, version something. And you can see that when this slide was written, is a while ago, it was CompuCell version 4.3.0.2. And we're now at 4.5.0.2. So we've got a couple of versions under the way. Okay, so we're going to open up our XML. Anybody need time for that, or does that work for people? Got it? Yes, no? Okay. So now we're going to scroll down through that. And I'm torn because I often will do this with a split screen, but if I split screen, the things get pretty small for the people who are looking at this from a laptop. So I'm going to try it this way. If people would rather have me do a split screen where I have Twit running at the same time, I can do that. So let me know how you feel about that. We'll try it this way for now, and if it doesn't work, I'll switch to split screen. If you scroll down through that XML is sort of semi-human readable. It's text, but it's not the most friendly format in the world. You will come, I think it should be about line 57, and you'll see something that will say steppable type uniform initializer. Do people see that? People find that? It may not be quite line 57 in your case, but you should have it there. Everybody got that? There in Twitter, there are search functions. You can do control F and it'll do a find if you want to search for uniform initializer. Okay. And within uniform initializer, this does the initial layout of the cell. It'll say region, box min, box max, gap, width, cell type, and region. And basically what this does is it draws a square where the bottom left-hand corner of the square is defined by box min x comma y, possibly z, and the top right is defined by box max. Width says that the cells are going to be drawn as 7 by 7 squares. Type means that there's only one cell type. In this case, we only have one cell type, but we could have random mixtures of multiple cell types. And gap allows us to say that when we draw our squares, we have a little bit of space between them if we don't want them to touch. Okay? And so now what I'd like you to do is change the box min and box max values so that we have fewer cells. And so you can go in and you can edit, make box min 40 instead of 20 for X and Y, and box max 60 instead of 80 for X and Y. All right, try that. And that's basically going to shrink the initial number of cells in the simulation. And we're also going to make our initial cell bigger. So we're going to make the cell 20 by 20. So there's only one big cell. Right? And so we'll change width from 7, which is the default, to 20. And so now drawing from 40 to 60 is 20. So we're going to get one big square cell. And then we're going to hit save all or save. Intuit it. And then you can go back to player and run it again. And so your goal here, all the steps are now here on the screen. Go through that and run the new simulation. Player should automatically update. So when you edit the file, when you restart it, it should load the latest version of the file. It should automatically update. If that fails, let me know and I'll tell you what. Then in that case, you may have to close player and reopen it from Twitter. So can people try that? And once you've done that, tell me what you got. Either in the chat or in person in the room. And again, if anybody needs help, please speak up. I'm here to help, and Pedro is also here to help. You have 9 cells in a smaller area. Did you change the width parameter to 20? I think you left the width as 7. So in instruction number 6, in the line that starts with width, change the value from 7 to 20. But this is the goal of this is to begin to. The worst thing that happens is CompuCell will crash, and you have to fix your simulation. Nothing dramatic will happen if you make a mistake. And so the goal of this is to start getting familiar and confident with how you begin to change the simulation to do what you want. And I realize that there are a lot of moving parts, and so it's not instantly obvious what to do. But once you've done it a few times, it becomes I hope becomes relatively natural. So Jonathan, when you change it to 20, did you get a single cell? 1 cell. Anybody else? That work? Anybody not get that? Everybody. Again, I don't want people to get bored, but I also don't want to lose people. So I want to make sure if everybody's in the room or everybody's online, it's a little easier for me to gauge where people are. When I have a split class, it's a little bit harder to do. All right. Okay, so we now made a cell. So did you get something that looked like that? Where it started out big and then it shrank? And the cell's probably pretty rough and scraggly looking, huh? If it happened too fast, you may actually want to use the step function instead of hitting run from the beginning. Hit stop and then step through it to see how it evolves one step at a time. Okay, did that work for people? Okay. So now let's actually here I managed to outsmart myself using Julio's slides. So I'm going to have to ask you to do something a little bit. Let's see, how do we do it? I'm going to get you back to where we need to be. Let's do this as an exercise. I would like you to go back and repeat what we just did from the beginning, which is open wizard, create a new simulation, create a single cell type, and now I want you to select an extra 2 boxes in that screen which says cell properties and behaviors. Select contact, which we did, volume flex, which we did, but also select surface flex and length constraint. That will generate a new simulation. And then you'll have to go back and do the same thing that we did just a minute ago, which is change the box min and box max and the width. So let's see if people can get that to work. And this time maybe I should do that in parallel with you so that we can have that together. Okay, so I'm going to go to CC3D project, new CC3D project, new simulation. I'll give it some random number for myself, 8. General simulation properties, rectangular slab, everything else the same. Next. 1 cell type, cell 1. Add. Next. No chemical fields. Next. And now I hit contact. That's the only one I did before. Volume flex. I'm going to actually add surface flex and length constraint. Okay. Does that work for people? And now I hit next. I hit finish. Here's my simulation. I'm going to open it in Player. People get something that looked like that? Is that what people are getting? Do people need me to go over it again? Or did that work for people? Okay. And now I'm going to go into Twit, go to my XML, scroll down to to get the size right. Scroll down to box min. I'm going to make that 60, 60, 80, 80. Width is 20. Save. And now I'm going to hit stop, step. And now I should have one cell. Okay. Were people able to get through that? Anybody have trouble with that? Jonathan, did that work for you? Santu, did that work? Santu is logged out. Did it work for
I'm
you?
changing the cell parameters, but yes.
Okay, great. Again, once you've done this once or twice, it becomes relatively quick to do it. The first 2 times you do it, of course, it takes a little more effort. I think you should be able to see everything you need on the one screen. The cell type is what the properties of the cell are, which we haven't yet really talked about.
Okay.
What's the volume constraint of the cell? What's the surface area of the cell? What's the target volume of the cell? Those are the things. And the reason that that's a little bit problematic the way CompuCell is written is that CompuCell groups things by the behavior. So all volumes for all cell types are specified together. All surface areas for all cell types are specified together. So it privileges the function, the process, rather than the cell.
Get it.
And so it's basically you're giving the same information, but the order in which you present things is not grouped by all. For cell type mesenchyme, I specify the volume, the surface area, and so on, so on, so on, so. Instead, I say the volume for each cell type is this. And so it's the hierarchy is different. Be easy enough to write a language where you specified it the other way, but then you'd have to write a little compiler to reorder it. And so that's something that we'd like to do. The place you have more problem is with what are called interactions, because the cell itself doesn't have an adhesion, because the way it adheres depends on what it's in contact with. And so it's not a property of the cell itself.
It's a property of the boundary.
Right. And that becomes, so those interactions are a little bit harder to. So again, we've picked contact energy, which we'll come back to a little bit later, the volume flex, surface flex, and length constraint. The 3 examples under constraints, volume, surface, and length, are these elastic constraints that we talked about already. And again, the dynamics try to satisfy those constraints. So we have 3 constraints. We have volume, surface, and length. And they all work similarly. And they all try to push the cell to have the properties that you've defined. So let's actually try to explore this. now. And we can do this again by going back to Twit. And let's look through Twit a little bit. And I'm going to make this a little bit smaller so we can see it all on one screen. If it's too small and you can't see it, let me know. And so let's look through our code. We have CompuCell3D, version 4.5.0.2 now. We have a block called metadata, which tells us essentially how to parallelize. CompuCell is intrinsically parallel. The parallelization is turned off, but if we have a fast CPU, we can tell it to use as many threads as we like, pretty much, with some limitations. And that works pretty well. Now we have this block called POTS that says we're on a lattice that's 256 by 256. We're running it for a long time. Fluctuation amplitude is 10. Neighbor order we didn't talk about, but that when we pick that target, when we pick a site and its target, we have to say how far the jump can be. That's what that is. We have a cell type list. We only created one cell type, cell 1 in this case. There's always a cell type medium. If we don't have anything defined, it's blank, that's medium. And then we've got a volume constraint here, a surface constraint, and a contact energy. And we have something called length constraint. And then the uniform initializer that sets up the initial condition. That's the simulation. If I look at the Python doesn't do anything in the simulation. It's blank. It's essentially empty. Okay. Everybody able to see that? And so now let's go in, and what we're going to do is we have all these constraints, and we're going to turn them on and off one at a time and see what that does to our simulation. And the first simulation, we only had the volume constraint. We didn't have the other ones available. This time we have them all there to begin with, and so we're now going to turn them off. So now, instead of having to redo the simulation each time, we'll turn the values of the constraints on and off. So we're going to now turn off the surface area constraint. And the way we're going to do that is, let's look at the surface area constraint. It's here. And we have surface energy plugin, surface energy parameters. It says cell type is cell 1. And then it says lambda surface, that's the lambda here, is 2, and the target surface is 50. So we could play with those two things and see what it does. The first thing we're going to do is turn it off. If we make lambda surface 0, then the energy contributed by that, the effective energy contributed by that, is 0. And so that's how we could turn it off without deleting those rows. So turn off the surface, and then go back and run your simulation again. Hit stop and step, and run it again. And you can ask the question, is it any different? Maybe yes, maybe no. But this is, you've now run the simulation with the volume, surface, and length turned on. And now you're running it with just volume and length turned on, not with surface. Okay. Okay. You always have to remember to hit save. In this case, if you closed player and opened it again, that would be different. Okay. And let's also turn off length constraint. And so we're going to go here. The order of your simulation components may be slightly different. Plug in name length constraint. Lambda length. You'll see lambda is the strength of each of these. Make that 0. Save. And now I go back to my simulation. Stop. and hit run. And there's my cell. Okay. So I'll give people a minute to get that working. And if you get stuck, there's an online version of it, but I hope people don't get stuck for this. Did that work for people? So now we're basically back to the simulation we wrote before the break, which is just the volume constraint by itself. Did that work for you? Okay, great. Anybody need more time? Okay, so you should see something that looks like this. Notice that the surface, if you zoom in, are people able to zoom into their results in the player? You can zoom in with a Control plus, or you can do under visual, here, plus zoom here. Depending on whether you're Mac or PC, it's slightly different to get zoom work. You can see your cell. And if we had looked more carefully, you would have seen that the first cell we had was rather scraggly on the boundary. That's because we told the cell to have a lot of surface compared to its volume, as it happens. In this case, we see a cell that's, quote, well behaved. It's fluctuating, it's not square, it's not too broken up. Do people get something that looks like that? Great. Good. Okay. So now we started out with the cell being 20 by 20, a volume of 400. Our target volume is 50. If we look at our target volume here, under volume target, here, volume, target volume is 50. Sorry, target volume is 50. All right. So now, why don't you change that target volume to be 400 and tell me what happens? So we're going to make target volume 400. Hit save. And then rerun the simulation and see what happens. What happened? Somebody tell me what happened. Before I do it, what's going to happen? Anybody? Or if you did it, what happened? Was the cell bigger, smaller, or the same as the before? Bigger. Fine. Okay, that's right. Now I have a nice big round cell. Now, there's a game you can play. What would happen if you set V target to 0? What's going to happen? Before you do it, what's going to happen? Somebody? Somebody online? Alex, if I make V target 0, what's going to happen to the cell?
Not sure.
Try it. Let me know. Target volume is 0. What happens? What's going to happen when I run it?
Will it collapse in on itself?
Yeah, that's right. Try it. I'm going to just do single steps here. Boop, cell dies. And that's actually a standard way to kill cells, actually, is to set their target volume to 0. They go shrink and disappear. Sometimes it's a little more complicated than that, but in general, that's what you do. Did everybody get that to work? Yes? No? Okay. If you make the target volume too small, then the natural fluctuations of the cell may cause the cell to go to 0 volume, in which case the cell will disappear. Once it's disappeared, it doesn't come back. So if the cell is small, you have to be a little bit careful about constraining it. Okay, if you change the lambda volume to make target volume 1,000, what happens? First try that, and then try changing lambda volume. See what happens. So let's make target volume 1,000. Save. Run it. Gonna get big here. All right. That's lambda, that's target volume. What happens if I make lambda volume very large? Make lambda volume 20 instead of 2. That's the strength of the constraint. And remember, the fluctuations depend on the ratio of the energies, which are proportional to the constraint strength. Is the volume of the cell going to be bigger or smaller now when I change lambda volume? The constraint strength. I've made the constraint strength bigger. Will the cell be bigger or smaller? Somebody? Try it. See what happens. Anybody tell me what they got? If people are lost, do say so, and I will go back and do things again. I don't want to leave people behind. Santu, did that work for you? Are you there, Santu? I do expect people to be on available to answer questions if they're online. How about Alex? What happened?
You make it really large, like it doesn't stops changing.
Yeah, it gets frozen, doesn't it? So the volume doesn't change, but the thing is going to freeze. Or at least the fluctuations will get smaller. If you make lambda volume very small, you'll see the fluctuations will get bigger, and you could even have the cell disappear if you make lambda volume too small. Where did my cursor go? All right. What if you make lambda volume 100? Let me show you something that which is a convenience that instead of editing the file, which we've been doing, there's something called steering, which is available inside of Player, which is the following. If you see here where it says Model Editor, if I scroll through Model Editor, I will see Plugin Volume. If I open Plugin Volume, it'll say Volume Energy. If I open that, it'll give me the same parameters that I defined in my XML.
So it's just like a preset thing or something?
It's a parse, this is the parse tree from the XML. The nice thing here is that I can change the values here as the simulation runs. So if I type 2 for lambda volume here and hit enter, and then hit run, well, let's see what happened. Why is it not? Ah, that's an interesting bug. I'll see the fluctuations are bigger. If I make lambda volume 200, it'll basically freeze it. Make it back to 2. So I can change these things on the fly. Now, the one thing about doing it that way is that you have to remember what you did. It's not logged. So if you make a change and you say, wow, that's really what I wanted, and you didn't write it down, It's not saved. Changing it here in the model editor doesn't change the
Just the
text.
permutation.
It's a dynamic. It's just it's a labile thing. The other thing that you have to be a little bit aware of in the model editor is that casting, Pythonic casting doesn't work. And so if it's 2.0 here and you want it to be 3, you have to type 3.0. If it's an integer, it's 1,000, and you want to make it 2,000, you have to type 2,000, not 2,000.5. So if it was an integer, you have to keep it an integer. If it was a float, you have to keep it a float. That's a little bit of an annoyance. It's easy. It's for integers, it's usually not a problem, but it's easy to forget if lambda volume is an integer that you have to type 2.0 because it was 2.0. Okay. So now you can play with it dynamically without having to stop it and rerun it. But I think you can see what happens. If I make the volume constraint very large, then the cost of moving away from the target volume is very high. And that means that the probability of accepting moves that are not exactly on the target volume is low. And so that reduces fluctuations. Another thing that I could do is keep the fluctuations the same, because what matters, as I mentioned, is the ratio of the fluctuation amplitude, which has this unfortunate name of temperature here, the fluctuation amplitude to the energies. And so I could change the temperature here, make it 10 times bigger, and that's going to make my fluctuations. Remember, it's in an exponential, so changing by a factor of 10 makes things not 10 times bigger, but e to the 10th bigger, a lot bigger. So if I make my temperature 100 and run it, why don't people try that and see what happens? What happens? Anybody? Now you could say, well, if I multiply the lambda volume by 10 and the temperature by 10, then the ratio has stayed the same. And that's true for lambda volume, but there are other energy functionals that don't get changed. So it's not quite scaling in the way you might expect. Let's try that. So here I have very high temperature, so the fluctuation amplitude is quite large. So I get a lot of fluctuation. Okay. In general, in our world, we're going to set that temperature value to be some number, typically 10, and we'll leave it. And we won't adjust that, but it's possible to do that if you want. Okay. So here you see we've taken the temperature, we've increased the fluctuation amplitude, and the surface of our cells gets fuzzier, get more fluctuations. If we go the other direction and we make that fluctuation amplitude too small, let's try it, make that fluctuation amplitude 1, what happens? Fluctuations will be less. If we make fluctuations too small, the simulation will freeze, and we won't get any movement at all. And so we're going to find that there's a range, usually a pretty big range, of fluctuation amplitudes and energies where things move fairly smoothly without falling apart. And if you see that your cells aren't moving at all when they should move, that typically means the energies are too small compared to the, or energies are too large compared to the fluctuations. If things are falling apart, then the energies are too small compared to the fluctuations. And that's actually something you get biologically as well. Cells do sometimes shed fragments of the cell. You get blebbing. That's often a pathological situation for cells, in cancer, for example. And there are also situations in which the cells, for example, are energy depleted and stop moving. So both of those things do occur, but they're not normal. So one wants to be aware of them. So we already did this example of making the temperature very small, and we saw that the cell basically got frozen. Let's look at the surface energy first, the surface area. So we've constrained the volume of the cell, and we turned off the surface area constraint. Surface area constraint basically is specifying the membrane area, the equilibrium membrane area of the cell. And in our case, we have to ask, how is the membrane surface area of the cell measured? And it's measured in a slightly strange way, which is we go around the surface perimeter of the cell and we count how many mismatched edges there are on the cell. So for example, if we had our cell was a single voxel, its surface would be 4. One mismatched edge, 2, 3, 4. If our cell were 2 by 2, it has 1, 2, 3, 4, 5, 6, 7, 8 surface voxels. Edges. And so its surface will be 8. And if we make it bigger, well, here it winds up that the volume is 21 and the surface is 20. Now, in normal Euclidean geometry, think about a circle. A circle has area pi r squared, and it's got a perimeter 2 pi r. So the surface goes like the square of the radius, and the perimeter goes linearly with the radius. On a lattice, that's not quite right when things are small. When you get big cells, hundreds of voxels, the relationship is linear. But when you have them be small, you get more surface than you would expect. And that's something we have to get used to. And that's actually, again, in the homework and exercise to try to work on that. So now let's change the surface constraint. So let us set V target to 1,000. Which we have here. Let's let lambda volume be 10 here. Let's set the contact energy, which we haven't really talked about yet. We're going to set that to 0. That lives here. Cell medium contact energy will make 0. And we're now going to set lambda surface to 10 and target surface to 126. That is, the surface is 4 times the square root of the volume. Try that. See what happens. We had set this to 0. We're now going to make lambda surface 10. And we're going to make lambda target surface 126. What happens? Somebody? Tell me what you got. Alex, did that do something for you?
I had to fix the temperature. I'll test it now.
Okay.
Good.
I mean, you can't go very far wrong. In other words, what we're doing is playing with the various parameters one at a time. If your temperature is small or large, you'll get slightly different results, but the relative things should still be similar. Anybody else? Jonathan, did that work for you?
I think so. I almost got a square with rounded corners.
Yep, that's right. It's basically because we don't have enough surface. So this is like blowing a lot of air into a beach ball. It gets very rigid. The thing that's funny, though, is that in reality, you would expect to get a circle, a sphere, but on a lattice, the smallest surface area you can have is a square. And so if you try to blow too much air into your beach ball, it gets cubical, which is a little bit strange. That's the lattice artifact. Did other people see the same thing? So now that you've done that, you can either edit the target surface here by making it bigger than 4 times the square root of target volume, or make it smaller. If you make it smaller, what is going to happen? If you make the target surface smaller than 126, what's going to happen? Is the cell going to be square or not square? It's going to be rigid or not rigid? Think about what you're doing. Think about the beach ball analogy. In the case of the beach ball that we have here, we've blown too much air into our beach ball, so it's very stiff. If we make the target surface smaller, we're basically shrinking the amount of rubber in the beach ball. We're trying to compress it even more. So we'll give it a shot. And then try going the other way. Instead of making the target surface 126, make it 256. And see what happens. What happens then? So somebody try it with target surface of, say, 50, and somebody try target surface of 256 and tell me what you get. And you can either do this again, you can edit this in the code that you got here, or you could do it in that model editor while it's running. Maybe it's more interesting to do it in the model editor while it's running. You can play with it more. Okay, so I'm going to go to Surface plugin. Let me hit pause for a second. Surface energy. First, I'm going to turn on lambda surface, make it 10. Target surface is 50, that's tiny. So what's going to happen? I seem to keep having to readjust the screen a little bit, it gets frozen. If I make target surface 300, that's not redrawing, that's my problem. If I make target surface 300, why is it not redrawing? This may be the bug that Maciek fixed in his new release. Okay, this is with target surface being small, and I get frozen. And now let me make target surface big. And it's not redrawing properly. That's something I have to apologize for. It's stuck. That's a bug. So much for having everything fixed, huh?
I think I saw the cell moving a little bit.
No, this is a redraw bug. Is it working for other people? This is definitely a bug in my install. So it seems to be fine. It seems to be fine when I do it with if I don't, okay, where is that stuck? Okay. I'm gonna try again. Interesting. Yeah, it's not redrawing the screen. Huh. It was working before. Okay, well,
That
SQLite file. I'm not going to do that. I'm going to let you guys play with it. If it's not affecting anybody else, it's not good. I'm not going to worry about it. What happened when people changed the value of S target? Did you see something that looked like this? If you make S target very small, you get a square. If you make S target intermediate, you get a circular cell. If you make S target very large, the cell falls apart. Why don't people try implementing those values and see what you get? See if you can recapitulate those examples.
When I set it to 9,000, it.
Dust.
And then when I tried to go back, of course, it wouldn't reform because each one was in its own.
Too spread out.
No.
Well, if you wait long enough, it'll condense. Yeah. It may take a while, but it'll condense. Did it?
Well, when it did that, I refreshed the simulation, so now I have to go back and
Okay.
set all the. Do you have the list of the parameters you're
Sure.
using for that? Thanks, guys. But the context parameters that need to be set.
It's sort of fun. You can actually get quite a bit. I mean, there's not that much you could do with one cell, but you could get sort of a reasonable variety of behaviors. There are cells that are pretty spherical and rigid. There are cells that are pretty floppy and extensile, amoeboid. So there are a pretty big range of these things that occur in real life.
Okay, so when I went to 5,000.
Yeah.
The dust.
Yeah.
And went back to 126.
Yeah.
They recondensed, but into 4.
Ah, yes, because they're freezing, and then once they're frozen, they can't. Okay, yes. If you move the target surface down very slowly, it'll come back together. I'm not recommending that one should do that as a matter of general practice in a simulation. It's interesting to explore the pathology. The other thing you could try if you're doing this and you're playing is set the volume constraint to 0 and see what happens if you only have a surface constraint. Generally, we almost never do that, but it can be interesting to see what happens with that. Surface, but no volume. Okay, I want to talk a little bit about another constraint that we have here, which we've got turned off, which is called the length constraint. The length constraint we don't use an enormous amount. By default, our cells are isotropic. They're basically round, modulo fluctuations. But in reality, cells sometimes are elongated. They have aspect ratios. And the length constraint constrains the aspect ratio of the cell. Now, for technical reasons, if you make the aspect ratio of a cell too long, you actually, what the length constraint is doing is actually constraining the moment of inertia of the cell. And the problem is that if I make the length constraint very large, the cell will split into two and then move the two blobs farther apart from each other, because that effectively is equivalent to being a long strut. And so then you have to play some games with things like connectivity constraint to say that the cell can't prevent the cells from breaking apart. But if you want an aspect ratio of something like 2 to 1, you can get it pretty reasonably using the length constraint. And so why don't people try that? Reset your values to your original values, and then turn the length constraint on. We'll turn the surface constraint off. We'll turn the surface energy back on. And now we're going to turn the length constraint on. Okay, try that. And you should, as you play with that, you should be able to pull the cell to make it more ellipsoidal by adjusting the target length of the cell. Is it clear what the exercise is, or is that not clear? And maybe somebody wants to screen share their result when they have something interesting. I'm always a little bit torn with this kind of, when I teach this. Historically, we've always jumped in and done things with lots of cells at the beginning. But when you have a lot of cells interacting with each other, it's a little bit hard to understand what the various things that you control the cell are doing independently of each other. And so it's not so exciting to spend a whole class playing with a single cell. On the other hand, once you've gotten their feeling for what one cell by itself does, then when you start putting them next to each other, it'll be clearer about what's going on.
I think it's better to do it this way. It's not riveting. But I kind of understand a lot more about the dynamics of this.
Well, and then also if there are bugs like the drawing bug in Player, it's easier to identify what the problem is. Not that there should be a bug in Player still after we just fixed it, but I think it's worth taking a time. Again, I'm using Julio's slides, which I think are very good. When I've done this way, I haven't had all the slides. I've basically said, okay, we're going to go into that model editor and we'll pull things. But I think there's some value in having it written down so that at least if you want to look back at the slide deck, you've got all the things there. Whereas if I just talk through it, yeah, you could recreate it yourself. Okay. So let's see what happens with length constraint. The length constraint, again, we have lambda length and target length. Let's try that. My instinct for volume and surface are very good because I use them all the time. I have to say, even for myself, the lambda length and target length are not things that I use that often. When you put the lambda length constraint in, what happened for anybody? 32 is our equilibrium. What if you make the target length bigger than 32? Make it 64. The discipline of the lecture is supposed to be we're going to turn everything off and do one parameter at a time. But of course, it's more fun if you change everything at once.
I mean that I do that and then I see what happens and I wonder, oh, if I change this, what would happen?
Great. That's exactly. I mean, the thing about CompuCell is modulo these annoyances that we faced with player and this. The point about CompuCell is it's designed so that you can play. And so especially this model editor function where you could, as it's running, you can change the value and see what happens. So you don't have to change the code base. You can play with it. Say, as I say, you have to remember what you did because it's not logged. Something we haven't looked at yet, which is what the output directories of these files look like. Whenever you run the simulation, it creates a new instance. And so it will actually save a copy of the code you ran. And then, in principle, if you do things like create movies, it'll save the movies in the same directory. So it'll bundle all of the outputs from the simulation with the code that generated them.
I did see that. There were like 10 directories from the last one.
What lambda are you using?
For the length constraint.
Yeah.
So the defaults were these. So I had surface constraint turned off here.
Lambda constraint. Where is it? Oh. Because it matters, right?
Sorry,
It matters.
which matters?
The lambda.
Well, here I haven't specified lambda length, right? Here I'm putting it back, so I'm setting things back the way they were. The fluctuations are 10, lambda volume is 10, value target is 10, lambda surface is turned off. Cell medium contact energy is defined as 10, which is where I started. And now I'm going to turn lambda length on with 5 and 32. And that should be what give me a round cell. That should basically be lambda length is satisfied when the cell is around. And so now you can define lambda length to be bigger than 32, make it 64 or something like that. And you should see an ellipsoidal cell. If you make lambda target length smaller than 32, I'm actually not sure what you'll get. You could try it. Okay?
I just get more circle below 32.
Yeah, I don't think it's going to do much because it can't. Fundamentally, it's going to be like making the target surface too small. It'll get stiffer. You should probably see fewer fluctuations. But I don't think it could actually. It can't make the cell the smallest that the effective target light could be is when it's round. And if you try to make it smaller, effectively you're compressing it. You're not doing, I think, anything else. But when you make it bigger than 32, does it give you an ellipsoidal cell? If you make it a lot bigger than 32, does it split into two pieces?
Yes, but I think if I increase the volume, I can stop that.
Another thing you could do is you could play with the target surface area, because when you split the cell into two pieces, the surface area is bigger than it would be if it was connected. So that's a game one could play too. But in general, you should see something like this. If you make the target length too big, the cell will split. And if you really want to make cells that are very elongated, like bacteria that are very rod-shaped, instead of trying to use this, what we do is we subdivide the cell into little compartments and do beads on a string. We would make, say, 8 little compartments and put them in a line, and then we could have the cell be as long as we want. That's a trick we won't get to till a little bit later in the class, but compartmental cells are a very powerful concept. Okay. Did that work for people? Do people have the basic idea of how to play with these parameters? Yeah, if you make the target length too small, then the cell could get frozen again. And you'll see this sometimes if you see simulations where things freeze up. Typically, that means that one of your energy functionals is way out of whack. It's not always obvious which one it is, but in general, when you see things freeze, that's a pretty good warning for that. This is a classic exercise. It's where the whole thing got started 30 some years ago, which is cell sorting. And I'll walk you through it and we'll see if we can get, it doesn't take that long to do. Exploring cell sorting fully can take a long time, but the basic ideas are not so complicated. We've seen the simulation before. Cell sorting is based on a very simple experimental observation, which is that you have in embryos, you build layered structures. A classic example of this would be the eye, where the pigmented retinal cells and the neural retinal cells form layers. And there's a hypothesis due to the late Malcolm Steinberg, which was in the next slide, called the differential adhesion hypothesis, that says that the layered architecture of tissues is based on the adhesivity of cells with each other. And this isn't totally crazy because cells have a whole variety of what are called cell adhesion molecules. And there are many species of cell adhesion molecules, neural cadherins, epithelial cadherins, endothelial cadherins, vascular cadherins, for example. And so you have both different types of cell adhesion molecules, and also different cells have different densities of them. And so that means that the adhesion energy, per contact energy between two cells, can be controlled very finely. You could have two cells of the same type stick to each other more than two cells of different type, or a cell likes to stick to a cell of a different type and not to another cell of its own type. And there's a lot of fine tuning that's possible. And there's a very simple set of experiments that you could do with embryonic cells that were done by Forgox and Foti working in Steinberg's lab at Princeton. Steinberg was a great man. He lived a long time, but I miss him. He was a great mentor, great inspiration to many people over a long period. And he did this experiment where he took embryonic cells. The reason people used eye cells was because in the 50s, when he started doing this work, fluorescent labels were not easily available. And pigmented retinal cells are black, so you don't have to label them. So they're really easy to see. And neural retinal cells are transparent. So you could build an aggregate, you could see the cells move around in it without doing any labeling. That's why he used retinal cells. And that's the experiment I did in my lab when I set up my lab. We'd take eyes out of embryonic chickens and then separate the cells. But the protocol starts out, remove 20 embryonic chicken eyes and then separate the cell layers. I used to do that experiment myself a lot, actually. So there was a day when I used to do mostly experiments, actually, in my life. Okay, so he takes different cells. This is the days when they had already color labels. And he takes, say, limb bud cells and epithelial cells. These wouldn't normally come in contact with each other. And you put them together, you could measure the surface energy between them, the surface tension between them. And you find that the more cohesive cells go to the middle, and the less cohesive cells go to the surface. And you could do this again and again. So limb bud cells are more cohesive than pigmented epithelial cells. And they go to the middle. Pigmented epithelial cells are more cohesive than heart cells, and they go to the middle. Liver cells are more cohesive. Heart cells are more cohesive than liver cells, so they go to the middle. And neural retinal cells are less cohesive than liver cells, so they go to the outside. And so there's this, and this pattern is that if heart cells are engulfed by liver cells, they're also going to be engulfed by neural retinal cells. So there's a transitivity of engulfment, which is not obvious. It doesn't have to be like that. You could imagine systems in which that wouldn't work. And so Steinberg came up with what's called the differential adhesion hypothesis that says that the sorting out of the cells is based basically on their contact energies, their surface energies.
Is contact energy and surface energy the same thing?
Yes.
Okay.
So it's a little bit complicated because we have a surface constraint.
Yeah, I know. I was trying to
And
figure
so
out.
that's why I try to remember to say contact, but in the old days we always said surface, so I may misspeak. So effectively, we're asking the question: if I bring two things together, two cells together that have a touch, there's some energy per unit contact, and that's the contact energy. And one thing you have to remember, which is always a bit confusing, is because if the things stick, the energy is negative. And so smaller energies mean more adhesion. And so the the hierarchy goes backwards. A smaller number means more adhesion. A higher number means repulsion. And so Steinberg's adhesion hypothesis said that cells type by type have consistent energy of contact per unit area with other cells of the same type but with different cells. And that adhesion depends on what kind of cell adhesion molecules they have and how many they have. And that the cells move randomly, fluctuate. And we're going to assume that they don't grow or change their properties during the duration of the simulation, and that more adhesive cells go to the middle and less adhesive cells go to the surface. And that picture actually there at the top is a picture that was done in my lab a long, long time ago by Zé Carlos, by my first Brazilian student, almost 30 years ago, 27 years ago, 28 years ago. And in this case, the black cells are, in fact, the pigmented cells from the eye of a chicken. And the transparent cells are the photoreceptors. They haven't differentiated fully yet, the neural retinal cells from the chicken. And you see, we start out with a random mixture, and the black cells clump up and gradually move to the center, and eventually you get a fully sorted aggregate. Now, there are a variety of other possibilities, in particular, that the contractility of the actin cytoskeleton varies between cells, and that's probably true. The cells could be sending out a chemical signal where black cells send out a signal that says, move towards me. That would be a chemotaxis hypothesis. And it's also possible that the cells actually send out very narrow filopodia, fiber-like things, anchor onto each other and pull on each other. And actually, all of these mechanisms, differential adhesion, differential contractility, coalescence from diffusion chemicals, and filopodial pulling, all of these things actually do occur in embryonic development. So as usual in biology, Occam's razor doesn't apply. Just because one mechanism could explain what you see doesn't mean that there's not another mechanism that does more or less the same thing that's also working. Because evolution doesn't evolve. There's no evolutionary constraint that says you can't do the same thing twice in two different ways. You may need to have it may, yeah, yeah. So Kyle says microbiome is like that too. So you have a lot of redundancy. Sometimes there are reasons to prune redundancy, but a lot of times you tolerate it because it works. In this case, in this particular case of the retinal cells moving around, we can actually show definitively that those other mechanisms are not in play, and that really, in this particular case, the only thing that's happening is differential adhesion. And the nice thing about differential adhesion is that we can do a thought experiment that explains what we see. So let's suppose we just have, to begin with, let's suppose we only have one cell type and medium. So suppose that we have two cells, cell and medium. Well, we have two contact energies. We either have cell touching medium. Medium touching medium doesn't make sense because medium is a single thing. But the cell can touch medium or it can touch another cell. Well, if the cell medium contact energy is bigger than the cell-cell contact energy, the pattern will have a lower energy if the cells are all clustered together. And if the cell medium contact energy is less than the cell-cell contact energy, then you'll have lower energy by having the cells disperse. So the most possible contact I can have is what I've drawn here as a checkerboard, and I can just count how many cell medium interfaces I have. 1, 2, 3, 4 for each one. So 1, 2, 3, 4, 5, 6, 7, 8. 8 times 4, I have 32 units of cell medium contact energy, and 0 units of cell-cell contact. So that's the most contact I could have. The alternative would be to put everything in a blob like this. I still have some cell medium contact, because no matter how I arrange it, there always is a yellow-green contact somewhere. It happens that I have a minimum of 12 surfaces. And if I count within that yellow blob, I have 10 units of cell-cell contact. And so now I can compare the two. Which is bigger? 32 times cell medium, or 12 times cell medium plus 10 times cell-cell? Well, I can just take the difference and then see which one wins. It's a little bit more work if I have 2 kinds of cells, but I could do the same thing. If I have 2 kinds of cells, I have contact between cell type A and cell type A. I have contact between cell type B and cell type B. I have contact between A and B. I have contact between A and medium and B and medium. So I have 5 contact energies. And the pattern is going to rearrange to minimize the contact energy. And that's what's happening here. In this case, I've defined contact energies so that my blue cells have a lower contact energy with each other than green cells do. And blue and green energy is intermediate between blue, blue, and green, green. And when I do that, I get the cells to sort. And since we're almost out of time, instead of having you develop the simulation, open CC3D project, go to demos. I can't do this for you, but you've done it before over at home. Go to demos and find cell sorting simulation. Does somebody want to, Pedro, can you screen share for me and show people where to find that? Here I have the old NanoHub version. I forgot to fix that NanoHub. Can I have you screen share and walk people through running that simulation? So let me stop the screen share. We'll turn things over to Pedro to close out the class.
Hello. All right. So let me go to edit. CC3D, oh sorry, CC3D project, open CC3D project. Then this is the installation folder of CompuCell3D on demos, and I think it's cell sort. This one.
Use book chapter demo. Use book chapter demos.
Okay, so book chapter demos, and then cell sorting. CC3D file. Double click, it opens. Let me close these ones. And here I can open in player. I already have a player opened.
Not the prettiest colors.
Yeah,
I want to make sure before we break, we're at the end of the class. I want to make sure everybody's able to run that demo. And then we'll keep working on that, get back next week. because the point is we spent today learning how to control a single cell. Now we're going to begin to think about how cells interact with each other. And the cell-cell adhesion is one of the simplest interactions that we can play with. And to be honest, you can get a lot with two cell types, and 5 adhesion energies. You can get one cell to disperse in the medium, the other cell to cluster. You can get both cell types to disperse in medium. You can get one cell to clump inside the other. You can get the cells to mix within the cluster. You can get a lot of different morphologies with the simple idea that you have two cell types and that they have contact energy in each other and in the medium. So you can do a lot of playing here. Pedro, you can show it off. You open up those plug-in energies. You can change those contact energies there. Try making the condensing, non-condensing energy big. Sorry, small. Make condensing, non-condensing small. So now smaller, remember, means more cohesive. So I make that contact energy smaller, the cells want to maximize their contact between each other.
Interesting.
So now they mix. Now make it bigger again. Now they start separating out. Why don't we make cell medium contact energy smaller? One of them. Just one. We make the medium condensing cells small, contact energy small. The medium cells are going to start moving out dispersing into the environment. Probably need to make it slightly smaller, like 3. And they're going to start spreading out. That's what happens in metastasis, by the way, where you lose cell-cell contact energy and you gain cell medium contact energy. So you can actually, this simulation allows you to explore a lot. And so next time we'll pick up with that. I hope that this was interesting for people. And I encourage people to play. There'll be a little homework assignment, which I'll probably send out tomorrow, which will try to consolidate some of the things that we did today in class. You see the cells now really are crawling out. You see the blue cells. The green and the blue are still separating relative to each other, but the blue cells, when they touch the medium, spread out. So you can get some really complicated morphologies just with these 5 parameters. And almost every possible combination exists in some biological situations. They're not almost every possibility of hierarchy of energies that you could have is observed. The sorting is a typical thing to happen. Dispersal and condensation is typical to happen. Mixing, that sort of checkerboard that you were seeing, is less common, and there are a variety of kinds of checkerboard. But for the famous example is Japanese quail oviduct. When females become sexually mature, the cells in the oviduct lining go from being mixed to being a checkerboard.